Now coming the last speaker, Safa, please.
Sorry. Okay, hello everybody, my name is Safa Jammali, I'm from University of Sherbrooke and I'm here today to present you our work on spliced alignment for the reconstruction of gene and transcript evolution. So, the current method working on gene tree reconstruction have a main approach which is based on 3 steps. The first, they refer each genes by one CDS, and usually the chosen one is based on the length. Usually it is the longest one. Here, for example, as you see, we have a CDS product for 3 genes, and the longest one are put in red. They are the longest one. And second, they compute the multiple sequence alignment from this chosen CDS, and finally they estimate their gene. But here, this approach has some limits. First, the selected CDS, or the chosen one, may be non-splicing orthologs, which means that they may contain some non-orthologous exons, which is the case here in the red. The red CDS and with the orange column, they are composed by non-orthologous exons. Second, the manner that they compute their multiple alignment may contain some errors. For example, they may align 2 non-orthologous exons in the same blocks, which is the case of the exons in the blue column. They are non-orthologous and they are aligned in the same block. So we propose our splice-aware approach, which make use of the splicing orthologs to further improve the gene tree estimation. So first, we compute multiple splice alignment to allow us to infer the splicing orthologs. Here, for example, we have multiple spliced alignment, and we infer the green CDS, which are the orthologs, and we can see that they are composed by orthologous exons. After, we deduce the CDS alignment from this multiple splice alignment, and finally, we estimate our gene tree. So the question here is how to compute the multiple spliced alignment. To do this, we start computing pairwise spliced alignment, and in which we use the sequence similarity, and we use also the splicing structure information. So here, when aligning, for example, 2 CDS to a gene, we take into account the sequence similarity, and in addition, we add the information of the splicing structure. So when you have a segment, we give more score, and we favorize a segment which has information about splice signals, splice site, exon junction, than a segment without these informations. And to do this, we have 3 steps. The first one is to do local alignment in order to identify preserved exon. And second, we do an extension of this gapped extension step in order to maximize the coverage and also to align the remaining exon. And we add the third step, which is dynamic programming approach, in order to align the remaining whole exons. And for each step, we use the information about the sequence and also the splicing structure. We extend our pairwise alignment to a multiple spliced alignment, and we want to maximize the sum of the score of induced pairwise spliced alignment. So to do this, we use a graph-based clustering algorithm. We want to detect connected equivalent exons, and once we have the connected exons, we make our multiple spliced alignment. Here we have some result of our spliced alignment. we compare multiple versions of our spliced alignment to current one, SP line, which is a current spliced alignment, SP line with light green, and the other color are the different version of our methods. And we use multiple data, real data and simulated data, and we variate the sequence similarity within the data. And we want to see the ability of each method, for example, to cover the CDS, to give an accurate alignment, to predict an accurate CDS orthology, and also to infer extremity at the level of CDS exon, gene exon, and canonical gene supply site. And globally, as we see, we have our method is robust, it is constant when varying the sequence similarity, but we see that SP line decrease when varying the sequence similarity. We also develop an interactive visualization tool in order to visualize the sequence of the multiple alignment and also to visualize the structure of the alignment. So our interactive visualization tool can or allows to highlight alternative splicing orthologs, potative exons, translation frame shift, and theirs, and also it allows to export sequences, region of sequences, and graphics. So what I want that to take home message for today is that we are working on two main projects. The first one is the spliced alignment, in which we develop a pairwise spliced alignment. We extended to a multiple spliced alignment, and we develop also an interactive tool to visualize the multiple alignment and to visualize the structure of the multiple alignment. And also we are working on phylogenetic reconstruction, in which we develop two main tools. The first one is a protein to gene tree, in which we estimate the gene tree based on place where clustering CDS, and the second one is super protein tree, in which we estimate the transcript phylogeny based on place where transcript. And for further discuss, you are welcome for the first domain in today is poster K44, and the second in poster K66 with my colleague Isai. At final, I want to acknowledge my two teams and all my sponsors to be here with you today. Thank you very much.
One question, please. Okay, so now we are closing this morning session, and we thank all this morning speakers again. Thank you. Thank you.